A very good morning to you all. Under industrial biotechnology, we have discussed about the general guidelines for the bioprocess design, the decision regarding the choice of the reactor, which is one of the most important component in the process design, and we have also discussed about the applicability of different types of kinetic considerations to different reactor types. In today's session, we will discuss on operation variables and parts of fermenter and also on recovery and purification strategies of fermentation process. Now the question is, what is fermenter? Fermenter is a vessel in which the biological reactions take place. The core biological reactions take place in the vessel, that is fermenter vessel. Whether chemical or biological reactions, it plays a significant role in the process of fermentation. In the graphical representation, you can see that the whole process has been built upon one core element. That means the entire fermentation process is dependent upon one core element and we call it as fermenter. Right. So this graph clearly shows that the whole process, if you are designing the process, then it has to be built upon one chosen core element and we call that core element as the fermenter. So thus any new element, when it is incorporated into the plant, the fermentation plant, it has to be based on the previous element, the data of the previous element. You cannot skip any of the step and you cannot change the order. So the order should be as shown in the graph. Right. So any new element incorporated into the fermentation plant has to be based on the data or the facts of the previous element. Thus, no step can be skipped and the order cannot be changed. Right. So in previous session we had discussed in detail about the differences between various mode of operations. Now we will try to understand the advantages and disadvantages of the reactor's mode of operation, be it in batch, continuous or fed batch mode. If we just look at the advantages in these three types of mode of operation, the equipment is simple, it is suitable for small production in case of batch mode, provides high production, better product quality due to constant conditions, and it is good for kinetic studies in case of continuous mode. Control of environmental conditions, that is substrate concentration as an example in case of fed batch mode. If you look at the disadvantage side, downtime for loading and cleaning, reaction conditions change with time in case of batch mode, requires flow control, culture may be unstable over long periods in case of continuous mode, requires feeding strategy to obtain desired concentrations in case of fed batch mode. So these are some of the important advantages and disadvantages of the reactor's mode of operation with respect to batch mode, continuous mode and fed batch mode. Now, moving on to the next component. The reactor should maintain a favorable environment. Favorable environment with respect to temperature, pressure, pH, mixing conditions, shear stress and media composition. So, the reactor should maintain a favorable environmental conditions with respect to temperature, pressure, pH, mixing, shear stress and media composition for the organism to grow. There should be uniformity and constancy of the parameters. The parameters are, as discussed, the temperature, pressure, pH, mixing conditions, shear stress and media compositions. Right. But it is not always possible to reach such a state in large scale or industrial scale operations. Right. So common operation variables which have to be supervised in different type of reactors are as shown in the next slide. So there will be common operation variables because it is not possible to reach such a state in real large scale or industrial scale operations. Now, what are these common operation variables? The common operation variables with respect to batch mode, continuous mode and semi-continuous mode is given in this slide. Number 1. Initial medium composition and inoculums. Number 2. Temperature. Number 3. Pressure. Number 4. pH. Number 5. Reaction time. Number 6. Aeration rate. Number 7. Stirring rate. So these are some of the common operation variables. That means these are the variables which are encountered during the operation of the fermenter or during the fermentation process, which are very much key in obtaining the product in desired quantity and quality. What are these common operational variables? Be it in batch mode, continuous mode or semi-continuous mode. Initial medium composition and inoculums, temperature, pressure, pH, reaction time, aeration rate and stirring rate. Right. Now let us look in detail about the different parts of fermenter and the associated functions. Number 1. Impeller or agitator. It is used for continuous stirring of media, distribution of the oxygen throughout the system and prevent cells from settling down. Number 2. Sparger or aerator. It is used to introduce sterile air or oxygen to the media in the aerobic fermentation process. Number 3. Baffles or vortex breakers. It provides better mixing of the cells by disrupting the vortex formation. Better mixing of the components and the cells by disrupting the vortex formation. Number 4, the temperature probe. It allows measure and to monitor temperature change in the medium during the process of fermentation. Number 5, the cooling jacket maintains the temperature of the fermentation process. Number 6, the pH probe. It is used to evaluate and monitor the change in pH of the medium. Number 7, the level probe. Most important because it maintains the desired level of the medium. So we have impeller, we have sparger, baffles, temperature probe, cooling jacket, pH probe and level probe. So continuing further, there is foam probe. So this detects presence of any foam in the fermentation process. Then we have acid, neutralizes the basic environment and thus helps in maintaining the pH. Then we have base, it neutralizes the acidic environment and maintains the required pH. Then we have anti-foam. So this is for breakdown and avoid any foam occurrence in the medium. Then we have sampling point to acquire sample throughout the process for testing. Then we have valves to regulate and limit the flow of liquids and gases. Then we have control panels to monitor all the set parameters in the system. Then there is inlet air filter for filtration of air before feeding into the fermenter. Then we have exhaust air filter, prevention of contaminants from escaping. Then we have a rotameter for measuring the flow rate of liquid or air. Then dissolved oxygen probe to measure dissolved oxygen in the fermenter. Then pressure gauge for measuring the internal pressure of the fermenter. So these are some of the important components or parts of the fermenter and its associated functions. Right. So after understanding thoroughly the principles and various mode of operation of the fermentation process, the recovery and purification of fermentation products is very much important. So before understanding in detail about the recovery and purification of fermentation products, we need to understand some of the important terminologies associated with recovery and purification strategies. Number 1, the adsorption. So this is a favored product binding to an adsorber or resin. Suppose if the desired product is in aqueous phase, so that it is a way of concentrating the product. That means favoring or allowing the product binding to a resin or an adsorber if the desired product is in aqueous phase and it is a way of concentrating the product. This is called as adsorption. Next we have chromatography. It is a unit operation for separation of the products from byproducts by passing through a defined column and then collecting the separated pure form of the product fraction. right? And then we have crystallization or precipitation. So it is one of the most popular and often used downstream process. What you are essentially doing is decreasing the product solubility by changing the medium or solvent composition and or solution conditions. So when we speak about the recovery and purification strategies of the fermentation product, these are some of the glossaries which are very important. Number one, adsorption. So this favors binding of the product to an adsorber or resin when the product is in the aqueous phase, and it is a way of concentrating the product. Then we have chromatography, wherein the product separation from the byproducts are done by passing the mixture through a defined or specific column and then collecting the separated pure form of the product fraction. And then we have crystallization or precipitation, one of the most popular and often used downstream processing step. And how do you do this? By decreasing the product solubility, by changing the medium or solvent composition and or solution conditions. Next we have distillation. That means the separation of more volatile products from less volatile products by way of evaporation and subsequent condensation. Then we have phase separation, the primary recovery step which makes use of separation of non-miscible phases such as solid, liquid, organic phase versus aqueous phase, or aqueous phase versus aqueous phase. Then we have membrane filtration. So it is the spatial separation of different molecules or products by polymeric or ceramic membranes of defined pore sizes. So what happens? This retain the products larger than the pore size and pass off the impurities, or it is vice versa. That means you can retain the product of interest and allow the impurities to pass off, or you can retain the impurities and allow the product to pass off. So it can be vice versa. And then if you pass off the product, you can collect the product at a later downstream process. So we have distillation, that is separation of more volatile products from less volatile products by way of evaporation and subsequent condensation. Then we have phase separation, that is primary recovery step which makes use of separation of non-miscible phases. Then we have membrane filtration. So it is the spatial separation of different molecules or particles by polymeric or ceramic membranes of different pore sizes. Right. So the knowledge of how to obtain the product from a mixture of many other undesired ingredients is a key factor for industrial biotechnology and biotransformations. So you need to have a thorough knowledge on how to obtain the product from a mixture of many undesired ingredients, which is a key factor for industrial biotechnology and biotransformations. The significant amount of work, energy and equipment that is needed for the separation of products, it is largely dependent on the type of the product you are separating. Right, whatever the amount of work, energy and equipment that is invested It is dependent upon the type of the product. Now, what is the type of the product that we are interested in? It could be a product mixture or highly purified product, a low molecular weight product or high molecular weight product, a stable product or highly labile product. Right. So the type of the product could be product mixture, highly purified product, low molecular weight, high molecular weight product, stable product or highly labile product. So whatever the amount of work, energy and equipment that is invested is dependent on these type of the product. That is not the only case. It is also dependent upon the type of the biological source, whether it is the source of the biomass, the crude extract, broth or fluid. Whether the source comes from biotransformation, microbial fermentation or cell culture, plant or animal tissues or body fluids. So the entire fermentation process has two variables: the type of the product and the type of the biological source used to obtain the product. So these two variables are key in recovery and purification strategies of the product. That is the type of the product, and number two, the type of the biological source. Whether the source is of the biomass, of the crude extract or of the broth or fluid, whether it comes from biotransformation, microbial fermentation or cell culture or plant or animal tissues or body fluids. The product recovery operations generally requires, number one, the isolation and purification of the product from the dilute aqueous media. So this is the basic step in recovery operation, that is product recovery operation. That means you should be interested to isolate and purify the product from dilute aqueous media, and often constitute the major cost contribution in the entire fermentation process. So it comes with a huge investment, a huge cost. That is isolating and purifying the product from the dilute aqueous media. So it is therefore very much important to develop high yield process with reduced amount of work and energy with minimal number of steps. So this is from industry point of view, because the isolation and purification of the product from the medium is associated with huge cost or a large cost contribution. So from industry point of view, it is important to develop high yield process with reduced amount of work and energy with minimum number of steps. If you just look at the overview of unit operations in the product recovery, we have the adsorption, distillation, chromatography, membrane filtration, recovery of solids and liquids, cell treatment, solvent extraction, liquid phase evaporation or separation, and crystallization and precipitation. So each unit operation is based on exploiting the unique physico-chemical properties of the product of interest compared to any other molecule present in the mixture. So there are several unit operations, right? but each unit operation is based on, so each, say for example, distillation, or it could be adsorption, or it could be solvent extraction, or maybe membrane filtration. It is dependent or based on exploiting the unique physico-chemical properties of the product of interest when compared with the other species in the mixture. The most important diversity that makes separation a considerable event is with respect to low molecular weight molecules and high molecular weight molecules. So first, there is a separation of molecules in terms of low molecular weight product and high molecular weight product. Okay. So if a fermentation broth is analyzed, when you start the process of harvesting the product, the specific product may be present at a low concentration. And it is not always the product that is present. It also includes intact microorganisms, cell fragments, soluble and insoluble medium components, and other metabolic products. So if you just analyze the fermentation broth during the time of the harvest of the product, you will find that it contains, along with the product which is supposed to be at lower concentration, so along with the product you find that the broth contains the intact microorganisms, cell fragments, soluble and insoluble medium components, and other metabolic products. So the product may also be intracellular. It can be heat labile, and easily it can break down by contaminating microorganisms. So all these factors tend to increase the difficulties in recovering the desired product of desired quality and quantity. Right? So to ensure good recovery or purification, the speed of operation may be the overriding factor because of the labile nature of the product. Therefore, the processing equipment or the fermenter must be of correct type and also of the correct size to ensure that the harvested broth can be processed within a satisfactory time limit. So with this information, I'll stop my today's lecture. We can continue our discussion on recovery and purification strategies in the next session. So thank you very much for your kind listening.